we will proceed to some scientific background information, and then we will open up for questions. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2016 ska tilldelas Yoshinori Ohsumi för hans upptäckter av mekanismer för autofagi. The Nobel Assembly at Karolinska Institute has today decided to award the 2016 Nobel Prize in Physiology or Medicine to Yoshinori Ohsumi for his discoveries of mechanisms for autophagy. So with that, I would like to leave the word to Professor Maria Masucci, who will describe a little bit about the science behind this prize. So please, Maria.
Okay. Yoshinori Ohsumi was born in Fukuoka, Japan in 1945. He started the seminal work for which he is awarded the Nobel Prize at Tokyo University, and he is currently an emeritus professor at the Tokyo Institute of Technology in Yokohama. This year's Nobel Prize in Physiology or Medicine is awarded for discoveries concerning autophagy. The word autophagy is derived from the Greek language and means self-eating, to eat oneself. In the mythology of many ancient human cultures, the self-eating dragon is a symbol of self-reliance and renewal. In the human body, self-eating, autophagy, is a process by which our cells use their own components to release energy and to build new functional parts. Autophagy was first observed in the 1960s, soon after the discovery of the lysosome, an organelle that contains the enzymes by the cells to disassemble proteins, sugars, and lipids to their usable building blocks. In addition to the lysosome, scientists observed another quite different type of organelle composed of a double membrane surrounding some cytoplasmic material. They also observed that vesicles appear in the cytoplasm of cells under stress, such as during starvation. The vesicles extended into double membrane that progressively engulfed a portion of the cytoplasm, forming what became known as an autophagosome. The autophagosome later fused with a lysosome, where the content was broken down by lysosomal enzymes. During the next 30 years, little more became known about autophagy. The mechanism for the process and the significance of autophagy for physiology or disease remain a mystery. The situation started to change in the 1990s, where Yoshinori Ohsumi decided to study the process of autophagy in baker's yeast. Yeast cells contain a single large lysosome known as the vacuole. Ohsumi found a clever way to visualize the accumulation of autophagosomes in the vacuole by making starved cells unable to degrade the content of the vacuole. He then used this method to search among thousands of mutants for yeast cells that had lost the capacity to activate autophagy. This ingenious strategy led to the discovery that autophagy is regulated by a large number of genes. As many as 15 essential autophagy genes were identified in the first study published in 1993. Having discovered the autophagy genes, Osumi moved on to the next question, that was to understand how the products of the genes work together. Thanks to this pioneering work, we have today a detailed understanding of the molecular mechanism of autophagy. Osumi showed that stress signals initiate a cascade of events that, through the formation of different protein complexes, control each step in the formation of the autophagosome, from the initial fusion of vesicles to the extension and bending of the membranes that sequester the cargo, and finally to the fusion of the autophagosome with the lysosome. Osumi and his colleagues were also first to identify genes that control autophagy in mammalian cells. This allowed the production of research tools that have transformed autophagy into the most intensely studied topics of biomedical research. Thanks to the work of Yoshinori Osumi and many others that followed in his footsteps, we now know that autophagy regulates important physiological functions of the cells. The effects of autophagy are associated with many human diseases. For example, autophagy is required for cell differentiation that gives rise to the many different types of cells that constitute our body. By recycling part of the cellular content, autophagy allows our body to cope with inflammation and with other types of stress. By capturing in viruses and bacteria, autophagy is essential for the body's defense against infection. Through the removal of damaged proteins and organelles, autophagy promotes cell renewal. Failure of autophagy is therefore associated with cell aging and with many diseases of old age, such as Alzheimer's disease and type 2 diabetes. Finally, too much autophagy may have undesired effects. In cancer, as for example in cancer, where autophagy promotes the growth of tumor cells and the resistance to anti-cancer drugs. The discoveries made by Yoshinori Osumi have been instrumental in revealing the mechanism and significance of a fundamental physiological process. And there is growing hope that knowledge will lead to the development of new strategies for the treatment of many human diseases.
Thank you very much. So before opening up for questions, I would just like to introduce, in addition to Maria Masucci, Professor Neil Larsson, and Professor Anna Videll, who's also the chair of the Nobel Committee, and they are here assisting to answer questions. So we'll open up for questions about the year's Nobel Prize. So, yes, I did. I was in touch with Professor Osumi this morning, right after the decision was made. He seemed surprised. The first thing he said was, I think he was actually not maybe expecting this, and he was surprised, but very pleased, and asked if he is allowed to leave the building. And of course, I explained that he would expect a lot of phone calls, and I think he was very, very pleased. So it was very nice to talk to him. It is 8 million Swedish kronor. Any more question? Here we have a question.
This is the journalist from People's Daily online. We are very happy that this year there is another Asian Nobel laureates, because last year was the Chinese Tu Youyou also got the Nobel laureates. So my question is, what's your comments on the research improvements in Asia in the medicine area?
Yes, I think, of course, the Nobel Prize is an international prize and takes no consideration to where anyone is from. And I think it is apparent to anyone in science that Asian science is growing enormously, not least, of course, in China. So I think maybe we're seeing the beginning of this. We should remember that the prize we're awarding today were for discoveries that were done in the 90s. So I think we can expect much more to come.
So it's going to be an Asian Nobel laureate next time also?
Not even we know
Not decided yet.
about that.
Thank you.
Was there up here?
Yes, my name is Annika Östman, Swedish radio company. What about Parkinson or cancer, or where do you think it is the most important contribution for the medicine?
I think I'll let Anna Vedell answer that question.
Okay, so can you hear me? Yes. So this is a basic discovery, but it's already clear that autophagy is critically involved both in the origin of disease and in our way of handling, counteracting diseases. And this applies to a broad range, such as cancer, neurodegenerative, Alzheimer, as Maria said, inflammatory diseases, diabetes. Exactly how this works in the different disease areas is what has been possible to study. So it has enabled totally new research areas now very active in addressing these questions. Neurodegeneration is definitely an active area, as is cancer. But the prize is for the basic knowledge and mechanisms and tools allowing these further research lines that are now exploding. So we can only hope for a broad impact in various diseases.
Okay, thank you. More questions? Here is one question.
In terms of applications, are there any concrete trials going on with drugs that are interfering with the system?
Lots and lots of activities, attempts to both simulate and correct autophagy, and it's a complex biology, but there's a lot of activities. And the specific way of really tailoring treatment is not really there yet, but it's really ongoing. Many clinical trials.
But no drugs on the market yet?
Well, there are drugs that affect autophagy, and also drugs where we didn't really know the mechanism and still don't. But they somehow affect autophagy. How this specifically work, we really have those drugs yet.
Maybe one can mention that most of the trials ongoing right now are for cancer. Did we see anyone more who wanted to ask anything? There are many eager for interviews that are welcome afterwards. So I think we can then probably conclude this press conference. Thanks so much for coming to Nobel Forum today. Thank you.
Ja, det är svenska. Och engelska vill ha på engelska. Eller till.
Are they coming now?
Professor Julien Sira, member of the Nobel Committee, could you please describe to us why our cells need to use autophagy or self-eating?
So autophagy, comes from the Greek terminology of self-eating. And without autophagy, our cells won't survive. We need autophagy to ward off invading molecules, for example, to deal with very large proteins that might be long-lived or perhaps defective. So we need to be able to have autophagy to degrade proteins, but we also need autophagy for renewal. In other words, we need autophagy to break down proteins for self-reliance.
What was the state of knowledge for the work of CS laureate?
So Yoshinori Kusumi started his work on this problem in the 1990s. And before that time, people understood that the cell, a lysosome, or in this case a vacuole in the yeast, people thought the vacuole was like a waste dump. People didn't know about molecules like the autophagy process, but you could see that morphologically existed in the cells. So the machinery was unknown, and how the system was working was unknown, and whether or not it was involved in disease was also unknown.
It's like a biological enigma.
Yeah, and I think Dr. Ashumi was interested in working on problems that other investigators stayed away from.
If you turn to the disease and the key findings, what's the first step here?
Dr. Ashumi worked with yeast cells, and they're a relatively simple cell. It's a singular cell. It's an organism with one cell. And he was interested in understanding things under the microscope. So the first problem he wanted to tackle was whether or not these yeast cells had autophagy. So he created a really ingenious way of studying them. He developed cells that didn't degrade proteins in the lysosome. And he reasoned that if he could stop the degradation process, he could see an accumulation of the autophagy machinery in these cells. So he looked at these cells under the microscope, and he subjected them to starvation that induces autophagy. And he could see an accumulation of phagosomes in the cell. So he could look at this under the microscope, and this machinery was moving under the microscope. You could see them visually. So that was the first step. Then, when he had a system where he could see an accumulation of autophagosomes in the cell, he could then study these yeasts by inducing a chemical that led to mutations. And he reasoned that if he could mutate the genes that were important for autophagy, he may be able to to then identify genes that regulated this process. So he could screen these yeast cells that were chemically modified, and he identified 15 genes that were important for the autophagic process for autophagy. He then went on, the third step, to clone the gene products, and he could study the proteins that were part of this autophagic machinery. He studied that this process was responsive to stress sensation. It involved a cascade of proteins that came together in complexes. And he studied these protein complexes and how the autophagosome was formed and how that was fused with the lysosome and how the contents of the autophagosome degraded in cells.
It seems to me this is a very complex system. In order to describe this prize in an accessible way, is there a metaphor one could use?
So I think you could think of early on the lysosome or the vacuole was seen as sort of a waste dump. And before Osumi came on the scene, people understood that that waste dump was in the cell. But what he showed was that it wasn't a waste dump, it was a recycling plant. This was a really sophisticated machinery that recycled damaged or long-lived proteins, and they were kind of reused. So if you think of this example, every day we need to replace about 200 to 300 grams of protein in our body. Every 2 to 3 months, every protein in our body turns over. Because of autophagy, these 200 to 300 grams of protein are made. We're eating proteins every day, about 70 grams of protein, but that's not enough to take care of the requirement to make new proteins. Because of this machinery, we're able to rely on some of our own proteins, maybe the damaged proteins or the long-lived proteins, and they're recycled with this sophisticated machinery so that we can sustain and we survive.
Also, this prize is for physiology or medicine, so would be the medical implications of these
Right, so
discoveries?
you're right that this award is for fundamental physiology. That's right. But it really has a very strong implication for different diseases. So, for example, we can think of with starvation. We need this process to survive starvation because we can have a self-renewal of our own protein so we can survive. That's one thing. We also need this process for embryogenesis. We need this process for cell division. We need this process to be functioning for normal physiology. There's examples of where this process is important to ward off diseases. For example, if you have a bacteria or a virus that infects a cell, we need this machinery to be able to ward off viral infections. The machinery is also important for diseases associated with aging. So, for example, if we have damaged or toxic proteins, they need to be handled. If they're not handled correctly, they can build up and lead to diseases like neurodegenerative diseases, Alzheimer's, Parkinson's, maybe diabetes. If you have too much autophagy, that may be associated with cancer, and the tumor cells may survive. So too much is not good, and too little is not good as well.
So if we turn to our attention to laureate, who is he?
So Yoshinori Osumi is still an active researcher today. He was born in 1945, so he's 70 years old. He just recently published a paper, and he's still working today on the cell biology and the structural biology of the autophagic machinery. So he's very, very active as a scientist, even yet today. He's a professor emeritus at the Tokyo Institute of Technology, of course, in Japan.
And finally, has the Nobel Committee been able to reach Professor Osumi?
Just this morning, Professor Perlman, the secretary of the Nobel Assembly, had a phone conversation. He got on the first number we had on our. He was in his office, in his lab, and I think he was very delighted. I think that he was surprised, wasn't expecting this, and he's looking forward to visit us in Stockholm.
Thank you so much.